Bismillahirrahmanirrahim. The synthesis of AMP and GMP. In the previous lecture, we have studied the biosynthesis of inosine monophosphate by the de novo synthesis pathway. This inosine monophosphate is the immediate precursor for the synthesis of AMP and GMP. We have earlier studied that both the purine nucleotides are synthesized from the inosine monophosphate. Now for the synthesis of adenosine monophosphate, first of all, aspartate condenses with the inosine monophosphate. In this condensation reaction, the GTP is required, and as a result, adenylosuccinate is produced. Then this product, adenylosuccinate, is cleaved and produce adenosine monophosphate. For the synthesis of GMP, this inosine monophosphate undergoes dehydrogenation, and this dehydrogenation process is catalyzed by the enzyme AMP dehydrogenase, which is an NAD dependent enzyme. As a result of this dehydrogenation, the AMP is converted to xanthosine monophosphate, and then in the next step, this xanthosine monophosphate reacts with the glutamine, and then the glutamine transfers its amide nitrogen to the xanthosine monophosphate to produce the GMP. So these reactions are elaborated in this diagram. Here we see that we have the precursor or the parent purine nucleotide IMP. This IMP reacts with an amino acid aspartate. This is basically the condensation reaction in which the whole of the aspartate molecule condenses with the IMP. This reaction is catalyzed by the enzyme adenylosuccinate synthetase, and this is the energy requiring process. For this purpose, GTP is utilized. So when this condensation takes place, the product which is used is the adenylosuccinate. Then the enzyme adenylosuccinate lyase or the adenylosuccinase, it cleaves the aspartate. As a result of this cleavage, the amino nitrogen of aspartate get attached to this IMP, and the carbon structure of this aspartate releases as a fumarate. So as a result of this reaction, this adenylosuccinate is converted to adenosine monophosphate. So this is a two-step reaction in which GTP is required for the synthesis of AMP. Now for the synthesis of GMP, this IMP, inosine monophosphate, undergoes dehydrogenation and is converted to xanthosine monophosphate. This reaction is catalyzed by the enzyme IMP dehydrogenase, and the coenzyme of this IMP dehydrogenase is the NAD. Then in the second step, this xanthosine monophosphate, it reacts with glutamine, In this reaction, the glutamine provides its amide nitrogen to the XMP, and after the transfer of amide nitrogen from the glutamine to the xanthosine monophosphate, the product produced is the guanosine monophosphate or the GMP. This reaction is a energy requiring process. In this reaction, the energy is provided by the hydrolysis of ATP. The enzyme catalyzed using this reaction is the amidotransferase. So we have seen in this conversion from IMP to AMP and IMP to GMP that for the synthesis of AMP, GTP is required, and for the synthesis of GMP, ATP is required. So in this way, both these nucleotides, ATP and GTP, they reciprocally regulate the synthesis of both these purine nucleotides. Now the second pathway for the synthesis of purine nucleotides is the salvage pathway. This pathway is basically the recycling of the purine bases or the purine nucleosides, which are provided by the degradation of the purine nucleotides within the body. The salvage pathways are of two types. One is the phosphoribosylation, and the other is the phosphorylation pathway. In phosphoribosylation pathway, the bases hypoxanthine, guanine and adenine are used as substrates which undergo phosphoribosylation. The other pathway, which is phosphorylation pathway, in this phosphorylation pathway, the purine nucleosides are used as substrates. In phosphoribosylation pathway, PRPP is the starting material which provides the phosphoribosyl group or the ribose 5-phosphate to the purine nucleotides. The free purines are salvaged by the two different enzymes. That is, the phosphoribosylation is catalyzed by the two enzymes. One is the adenine phosphoriboside transferase and the other one is the hypoxanthine guanine phosphoribosyl transferase. This is the single step process which is catalyzed by the adenosine phosphoribosyl transferase enzyme, in which the free adenine reacts with phosphoribosyl pyrophosphate to yield the corresponding adenine nucleotide. The free bases guanine and hypoxanthine, these are also salvaged in the same way, and the enzyme catalyzing their salvage is the hypoxanthine guanine phosphoribosyl transferase. In this reaction, the phosphoribosyl group is provided by the PRPP, phosphoribosyl pyrophosphate. This phosphoribosylation process is especially important for some tissues like RBCs and brain, because the de novo synthesis does not take place in these tissues. Another importance of this salvage pathway is that it economizes the intracellular energy expenditure. That is, in this salvage pathway, very low amount of ATP or GTP is required, which saves the energy. This is the diagram which shows the salvage pathways, the phosphoribosylation of the bases adenine, hypoxanthine and guanine. The adenine, it reacts with phosphoribosyl pyrophosphate PRPP. In this reaction, the PRPP provides the ribose 5-phosphate, which is transferred to the adenine base, and after cleavage, this PRPP from this PRPP pyrophosphate group is released, and ribose 5-phosphate is attached to the adenine to give rise to the adenosine monophosphate, and this reaction is catalyzed by the adenine phosphoribosyl transferase. The other purine nucleotide, the other purine base, the hypoxanthine, is also phosphoribosylated by the PRPP. In this reaction, the hypoxanthine attaches to the ribose 5-phosphate to form the IMP, inosine monophosphate. Similarly, the another purine base, guanine, it also reacts with PRPP and is converted to GMP, guanosine monophosphate. Both these reactions are catalyzed by the enzyme hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase. So this is the pathway for the phosphoribosylation of the purine bases. Now the another type of salvage pathway is the phosphorylation of nucleosides. As we know that the nucleosides have two components, one is the purine base and the other is the ribose sugar. So for the synthesis of for the conversion of ribonucleosides to ribonucleotides, only phosphorylation is required. So for the phosphorylation, the phosphate group or the phosphoryl group is provided by the ATP, and the enzyme catalyzing this reaction is simply the kinases, ribonucleoside kinase. In this reaction, we see that this is the purine nucleoside, in which this is a PU is the purine base and R is the ribose sugar, that is the nucleoside base, nitrogen base plus sugar is the nucleoside. It reacts with ATP. This ATP provides the phosphoryl group to this purine nucleoside. You can see this purine ribonucleoside with P, that shows the phosphate group. So this is purine nucleoside is converted to purine ribonucleotide, and ATP is converted to ADP. So these are both the salvage pathway for the conversion of purine bases and purine nucleosides into the purine nucleotides. Now there are some synthetic inhibitors which can inhibit the synthesis of purine nucleotides. These are the mercaptopurine, mycophenolic acid, azacitidine and diazonorleucine. And the other type is the antifolate drugs, which are methotrexate and sulfonamides. The numbers shown in this diagram, that is 13, 14, 14, 5 to These are the numbering of the reactions which are given in the book Harper's Biochemistry. If you see the diagram of de novo synthesis of purine nucleotides from the book Harper's Biochemistry, the reactions are numbered. These numbers shows the reactions which are inhibited by the respective synthetic inhibitor. The antifolate drugs, which is the methotrexate and sulfonamides, this methotrexate is the inhibitor of the tetrahydrofolate reductase. This enzyme converts the oxidized form of tetrahydrofolate to the reduced form of tetrahydrofolate, which is then used for the synthesis of purine nucleotides. So the methotrexate inhibits the reduction of tetrahydrofolate with reduction of tetrahydrofolate, which is then used in the synthesis of purine nucleotides. So this methotrexate, when inhibit the oxidized form of oxidized form of tetrahydrofolate into the reduced form of tetrahydrofolate, then it inhibits the synthesis of purine nucleotides. Similarly, the sulfonamides are the structural analogues of para-aminobenzoic acid. These sulfa drugs are used to inhibit the synthesis of folic acid in the microorganisms. As we know that the synthesis of folic acid takes place only in the microorganisms, the humans cannot synthesize the folic acid in their bodies. So the sulfonamides have no influence on the human cells. They only affect the synthesis of nucleic acids like DNA and RNA only in the microorganisms. Another synthetic inhibitor is the mycophenolic acid. This mycophenolic acid basically inhibits the enzyme AMP dehydrogenase. So the conversion of AMP to GMP. So the mycophenolic acid, it deprives of the T and B lymphocytes of the key components of nucleic acids. So with the depredation of GMP and then the AMP, the nucleic acids are not synthesized. As basically this drug mainly affects the T and B lymphocytes, so it inhibits the immune responses. Thus this drug is used as an immunosuppressant, especially for the prevention of graft rejection reactions. Now we move to the regulation of the purine nucleotide biosynthesis. The biosynthesis of purine nucleotides is basically regulated at 3 steps. The most important one is the conversion of PRPP to phosphoribosylamine. This reaction is catalyzed by the enzyme PRPP glutamyl amidotransferase. This enzyme is inhibited by the nucleotides AMP, GMP and IMP. Similarly, Other important step for the regulation of synthesis of purine nucleotides is the conversion of ribose 5-phosphate to PRPP, that is the synthesis of PRPP, and this enzyme is catalyzed by the PRPP synthase. So this enzyme PRPP synthase is regulated by ATP. The third step of regulation of the synthesis of purine nucleotides is the conversion of inosine monophosphate to AMP and GMP. In this diagram, it is clear that the conversion of IMP to GMP is inhibited by feedback regulation, negative feedback regulation by the product GMP. And similarly, the synthesis of AMP is inhibited by the product AMP. This is also the example of negative feedback inhibition. As we have seen earlier, that for the synthesis of GMP from IMP, ATP is required, and for the synthesis of AMP, GTP is required. So both ATP and GTP, they reciprocally regulate the synthesis of both nucleotides. That is, one nucleotide controls the synthesis of other one, and the vice versa. Now this is the summary of the regulation of purine nucleotide biosynthesis. We briefly see that the one enzyme is the PRPP synthase or PRPP synthetase. This enzyme is activated by inorganic phosphates and is inhibited by the purine nucleotides like AMP, GMP and IMP. Another committed step is the PRPP glutamyl amidotransferase. This enzyme is activated by PRPP and glutamine and is inhibited by AMP, GMP and IMP. Thank you.